Hello everyone. Today we are going to discuss about the DNA replication and DNA mutation. First we are discussing about the DNA replication. We know that the DNA is a self-replicating structure and the replicate semi-conservatively. The DNA replication is the catalyzed by the set of the enzymes. In this process of the DNA replication, the DNA makes the multiple copies of itself, and it is a biological polymerization which proceed in a sequence of the initiation, elongation, and the termination. The DNA polymerase is the main enzyme in the replication process. The first is initiation. In the DNA replication demand a high degree of the accuracy because even a minute mistake will result in the mutation. Thus, the replication cannot initiate randomly at any point in the DNA. For the replication to begin, there is a particular region called the origin of the replication. The replication begins with the spotting of the origin followed by the unwinding two DNA strand. The unwrapping of the DNA strand in the entire length is not feasible due to the high energy input. Hence, the first replication fork is created catalyzed by the helicase enzymes which 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 are the help the DNA strand. In next process is elongation. At the strands are the separated, the polymerase enzyme start the synthesizing the complementary sequences in each of the strands. The parental strand will act as a template for the newly synthesized daughter strand. It is noted that the elongation is the unidirectional, that is the DNA is always polymerized only the 5' to 3' direction. In one strand it is continuous, hence called as a continuous replication. A continuous replication, while in other strand, that is 5' to 3' template, it is continuous replication. They occur as a fragment called as a Okazaki fragment. The enzyme DNA ligase join them later. Next one is the termination. The termination of the replication occur in different organisms. In E. coli, like organisms, chromosomes are the circular, and this happen when two replication fork between the terminal meet each other. The replication is the semi-conservative. The parent DNA has the two strands that are the complementary to each other, and both strands undergo the simultaneous replication, which produce the two daughter DNA, and have the half of the parent DNA and half of the new DNA. This type of the replication known as the semi-conservative, and because of the half of the original DNA is conserved in the daughter DNA. The first experimental evidence provided by the Michelson and the Shah in 1958. Next, in DNA replication, DNA carries the genetic information from the generation to the next generation. So the regarded as the reserve bank of the genetic information or the memory bank. The DNA transfer the information to RNA translation information, then it transfer to the protein synthesis. Now let's see about the DNA mutation. The DNA mutation is a change to the gene's DNA sequence to produce something different, and it create the permanent changes to the gene's DNA sequences. The type of the mutation which are on the basis of the where they are located. The gene mutation, that is frame shift mutation and the point mutation. The point mutations, in point mutation there are mainly three types, that is substitution, insertion and the deletion. First, substitution. During replication, one base is the inserted incorrectly, and replacing the pair at the appropriate location on the complementary strand. Example of the substitution mutation are the sickle cell anemia. Next one is the insertion. In the replication, DNA, one or more additional nucleotides are the added frequently, causing a frame shift. Example, one form of the beta thalamus. Third type is the deletion. During the replication process, one or more nucleotides may be skipped or removed, which usually causes a frame shift. The example is a cystic fibrosis. Next is a chromosomal mutation. In a chromosomal mutation, inverse ion deletion, duplication and the translocation is processes takes place. First, we are seeing the inverse ions. In this, the flipping and the reinsertion of the single chromosomal region takes place. In the deletion, next one is a deletion. In deletion, when the chromosome segment is lost, all the genes in that segment are also gone. And next one is a duplication. In duplication, a chromosomal segment is repeated, increasing the concentration gene in the area. Example are the cancer. And in the translocation, a section of one chromosome is inappropriate, which is joined another chromosomes. Example is leukemia. The copy no variation gene in that gene amplification. In gene amplification, an increase in the made of in the tandem copies of the locus. Causes of the mutation. The mutation in the gene coding for the HB causes the sickle cell anemia. First cause is the internal. The internal cause, most of the mutation occurs when the DNA fails to copy the accurately. The example are the lifestyle. Because of the lifestyle, it can also affect the gene mutation. Then what type of the food we are taking, smoking and the chemicals we are using day by day. Next is the external causes. When DNA is exposed to the certain chemicals or the radiation, it causes the DNA to break down, and the UV radiation cause the thymine alter to break the resulting muted DNA. Example are the PCR and electrophoresis. And third cause is the spontaneous. In spontaneous, those that are the result of the natural changes in the DNA structure, that is genetic factor. And last one is a